Alright, so today we're going to be talking about recombinant DNA. Now this is a little bit different than things that we've already talked about because in this circumstance we're actually going to be manipulating our DNA to make it form things that we need it to and things that we can apply to in our everyday lives. So first off we have to talk about, well, how do we get this? And this is through genetic engineering. So we are engineering genes in terms of speaking. So this involves cutting or cleaving DNA, same thing, from one organism into small fragments. So we all know how long these DNA molecules are. These are going to be cutting them up into small fragments that we can take and use in a different kind of situation. Now into small fragments and inserting those fragments that they take from cleaving that original DNA into a host organism of the same or different species. So for instance, one that I will probably reference a lot throughout this entire lecture is going to be a tobacco plant that they actually took the DNA from a firefly and they took the DNA that made that firefly glow and they actually put that into a tobacco plant in order to make that tobacco plant glow. This is one of the applications of recombinant DNA. Now recombinant DNA, the reason that it is called that is because it's made by connecting or recombining fragments of DNA from different sources. So you can take all these different sources of DNA, combine them all together, and put them into a host organism to cause that host organism to exhibit all different types of characteristics. Be resistant to pesticides, for instance, or be resistant to a certain type of bug, things of that kind of nature. So many practical applications. Now, if you do this and you do put recombinant DNA into a different organism, you're going to end up with what is called a transgenic organism, which is plant or animal that contain functional recombinant DNA, and that's the key. It has to be functional. It can't just be kind of in there where the DNA didn't really take. It has to be functional, has to be contributing that characteristic or that extra DNA into that host organism, contain functional recombinant DNA from an organism of a different species. Okay, so again, example, your tobacco plants that glow from your firefly DNA. Now, first thing, how do we make recombinant DNA? There are 3 steps. Our first step is that we have to isolate the foreign DNA fragment that we want to take from that original organism and be able to transplant it into a host. Second step, we have to attach the DNA fragment to a carrier. We can't just take the DNA out of one organism and then go and place it in another and expect for it to be able to take and for it to be able to replicate. We have to attach it to some kind of carrier to make sure that that carrier, once planted in the host organism, will then be able to make sure that that DNA replicates and continues on its journey. And third step, transfer it into the host organism. We have to actually finally put that in. That will be our last step, is putting it into our host organism of choice. For example, again, we would take that firefly DNA, put it on our tobacco plant, and our tobacco plant would be our host organism. So your first step, isolating the fragment. So we have small pieces of DNA that have to be cut from each chromosome. That is made possible by the discovery of restriction enzymes, which were discovered back in the 1970s. So restriction enzymes kind of do exactly what they sound like. They restrict that DNA to the point where it is restricted so tightly that they cut off the DNA. So that is their job. So whenever you think about restriction, think about it restricting so tight that eventually it actually cuts off that DNA. So restriction enzymes are bacterial proteins that can cut off or can cut both strands of the DNA molecule, so both strands, both sides, at a specific nucleotide sequence. So that's the key. These restriction enzymes are looking for that specific nucleotide sequence, such it might be any given number of things. There are restriction enzymes for many different types of nucleotide sequences, pretty much anything that you can think of. Let's say that ours is looking for ACCG. Whenever it comes across that ACCG sequence in our DNA, it's going to cut that DNA at that specific sequence. So once again, bacterial proteins cut both strands of the DNA molecule at a specific nucleotide sequence. And there are many different restriction enzymes. Pretty much any kind of combination that we can have in DNA, we can find a restriction enzyme to cut that DNA with. Now, big number 1, cutting DNA is only possible with restriction enzymes. We can't take in a small pair of scissors and go cut that DNA into as many different pieces as we want. We have to rely on those restriction enzymes. Thing is that we also have to realize about our restriction enzyme is that based on the nucleotide sequence that they are looking for to cut, might cause our DNA to be different strands or different lengths. So we might have a nucleotide sequence closer to the beginning of our DNA strand that is being cut by that restriction enzyme, but then we also might have it somewhere in the middle. That can result in different lengths. Alright, some DNA sequences are the same on both sides of the strand. So both sides are the same, both strands are the same, only in opposite order. So these are called palindromic sequences. So whenever I say palindromic, think of the word palindrome. Palindrome means anything that's spelled the same backwards and forwards, like race car, mom, dad, Hannah, things like that. Those type of words are palindromes. So again, your palindromic sequences, same thing, front and back, just opposite order. Okay? Now, some enzymes cut these palindromic sequences by unzipping them for a few nucleotides. So just like we had with our transcription whenever our DNA was cut into two or unzipped, basically the same thing is going to be happening here. These enzymes, those restriction enzymes, cut those palindromic sequences by unzipping them. We can also have other enzymes, other restriction enzymes, produce fragments in which DNA is cut straight across both strands. And then these are going to be known as blunt ends. Think of just like if you cut a piece of paper with a pair of scissors, you just cut it straight across. It's not going to be sharp, it's going to be pretty blunt, it's not really going to do a whole lot of damage. Same thing that we have for our DNA. In either case, however, when these double strands are cut, they end up with single-stranded ends. So you can think of it like it's cutting not the entire strand all the way across in a perfect straight line, but it might be cutting them kind of at a diagonal. If it does that, we're going to end up with those single-stranded ends. Now, these single-stranded ends attract other single strands to bond with them. So for this reason, they are called sticky ends. So they are being very sticky. They are attracting these other forms of DNA, these other single strands of DNA, and essentially they are binding with them. Now, these sticky ends are the key to recombinant DNA. And the reason for that is going to be because whatever restriction enzyme has cut out that one nucleotide sequence, it's done it somewhere else too, which means that they will have the same DNA sequence and they will be able to bond together. One sticky end that the same restriction enzyme has cut will bond with another sticky end that the same restriction enzyme has cut, come together and form that entire DNA sequence. But now that DNA sequence has been recombined, forming our recombinant DNA. So which is why they are the key to recombinant DNA. If we did not have these sticky ends, recombinant DNA would not be able to work in the way that we need it to. Alright, the second step. How do we get these into our host organism? So we call these vectors. Vectors will transfer DNA. So loose fragments of DNA do not readily become part of a host organism's DNA. That host organism will sometimes try to reject that DNA if it's not attached to anything. So we have to use vectors. So vectors must be used. A vector, this is the means by which DNA from another species, like that firefly, can be carried to the host cell, into the tobacco plant. So these may be biological or mechanical. So the ones that we're going to talk about a plasmid. That's going to be our main biological vector that we're going to talk about. It's a small ring of DNA that's actually found in a bacterial cell. So again, this is a biological vector. They will put in that recombinant DNA into that DNA of the plasmid, and it will replicate itself. And we're going to talk about how it does that. They can also use a micropipette. Now, a micropipette is similar to the pipettes that we use in class. However, it is much, much smaller and much, much more precise in what they're doing with it. It would be inserted into a cell, inserted into the nucleus, and they would inject that pipette, or they would inject the DNA into the cell. The last type that we're really going to concern ourselves with is a microscopic metal bullet, essentially, coated with DNA. They've coated it with DNA. It's shot into the cell from a gene gun. So similar to the micropipette injecting it in, similar to a needle. Same kind of thing, except this is going to be much more dynamic and much more quick. And it's also going to be, it can damage the cell in some other ways that, say, micropipettes cannot. Right, and the foreign DNA is recombined. Whenever that foreign DNA, we take that foreign DNA out of our cell, or out of whatever cell we're trying to get, for instance, the firefly DNA gene that makes the fireflies glow, is recombined into a plasmid, or viral DNA, we can have either one, with the help of a second enzyme. So they take that DNA out of that original cell that they want, they put it back into the plasmid, and that's going to be processed by a second enzyme. Now, the third step, how do we insert that into our host? So after the DNA is inserted into the plasmid, we've put that into the plasmid, which remember, the plasmid is just its own small ring of DNA. After the DNA, the recombinant DNA, the DNA that we want from our original cell, like our firefly gene, is inserted into the plasmid, it is transferred into a bacterial cell. So bacterial cells can replicate themselves very quickly. So this plasmid in this bacterial cell, in this one bacterial cell, can produce up to 500 copies of DNA per bacterial cell. So taking from one copy of DNA that we have got from our original cell, like our firefly gene, now after we put that into our bacterial cell, we can now produce up to 500 copies of it. Now those copies, each identical recombinant DNA molecule, is called a gene clone. So it is the exact same copy of itself, is a clone of those genes. And plasmids can also deliver genes to plants or animals, and every time the host cell divides, the recombinant DNA is cloned. So if we don't want to put it into a bacterial cell, say for instance, we just want to take our plasmid, which contains our DNA gene that we want, like our firefly gene, we might just put it directly into our tobacco plant or directly into our host cell in our host organism. Every time that host cell goes through the process of mitosis, it will replicate that DNA, it will divide, and we will see the recombinant DNA will be cloned once again. In order to replicate DNA outside of living things, however, we have a couple options. In order to replicate DNA outside of a living organism, a method known as polymerase chain reaction, or PCR, is used. So they might choose to use this if they need millions of copies of DNA present in a very short time span. PCR is actually capable of cloning millions of copies of DNA every single day. So this is your recombinant DNA lecture. This is your recombinant DNA notes. Good luck.